Hi, we're MyBiome, and today we're going to present to you what we've done throughout this semester with our work on microfluidic devices.
Hi, my name is Francisco Cepeda.
I'm Ravalika Demerla.
Hi, I'm Margaret Sang.
I'm Manupala Parta.
And we wanted to pay tribute to a dude who couldn't be with us right now, but she was a really integral part of the work that we did at the beginning. Okay, so what we did this semester is try to create an in vitro gut-on-a-chip system, and one of the main applications of this system is to be able to do personalized medicine. And so gut-on-chip devices, or in general organ-on-chips, the idea behind them is that you can take the cells from any patient, you can take their kidney cells or their liver cells or anything like that, put them into these devices, and then conduct different assays. So for example, drug metabolism assays, to see how individuals' cells respond to a specific treatment. And that kind of idea can create personalized medicine, and that's sort of the goal we were trying to go for with this in vitro gut-on-a-chip system. And so when we were looking through past literature, one paper that we found that created a very simple but effective gut-on-a-chip device was created by Guo et al. And essentially they had these 4 channels where they were able to culture cells. They had a membrane underneath and then a glass slide. This was a PDMS-based device, but it was very simple, but it was still effective. They had a peristaltic pump, so you could create peristalsis, which we all know is very important to how the gut works. And so this paper is kind of the inspiration for what we decided to do moving forward. And so also when we were looking into literature, we found that there were a lot of difficulties that have been conceived while trying to create these realistic gut-on-a-chip devices. For example, it can be difficult to culture human intestinal epithelial cells, and also trying to co-culture with other critical intestinal cell types and bacteria is very difficult, but it's also very important because, as we all know, in the gut there are many different species of bacteria and cells living that all have some kind of crosstalk, which is really important to be able to model in these in vitro systems. We also found that it can be difficult to mimic intestinal physiology. For example, the microvilli structure that is present in all of our guts is difficult to create. And also creating realistic flow conditions and doing things like peristalsis. That was also kind of an area where people have difficulty sometimes doing. And then also trying to create this multiple organ crosstalk. So if you have a gut-on-a-chip device, you also want to be able to interface that maybe with a brain-on-a-chip device, because there's a lot of crosstalk between your brain and your gut. And we also see that drug assays can be difficult to conduct on PDMS-based devices. And so PDMS, which is the standard for microfluidic devices, has a lot of benefits, but it also has some limitations. One of them being that it absorbs hydrophobic materials, which makes it difficult to perform reliable drug testing. And the NIH is no longer going to fund PDMS-based devices. And something that we noticed is that it's quite easy to fabricate with PDMS-based devices. If you can 3D print a mold and then fill that mold with your PDMS and create your device, and then you can just insert your inlets and then do some plasma treatment and attach your PDMS-based device to a glass slide. And it's a pretty simple fabrication process. So we wanted to try and create something similar to that, but without using PDMS.
So initially what we decided to do is we decided to use a material called SEBS, which stands for styrene, ethylene, butylene, styrene. And basically the key characteristics of SEBS that we thought were really important is that it's wettable. The most important one, I think, is that it doesn't absorb hydrophobic molecules, which PDMS does, and it has a high bond strength. So initially what we planned on doing was using the same model of the microfluidic device that was in the Glow paper, but instead of using PDMS, we would use SEBS as our material. So while designing SEBS, so we first created a mold, and one thing that we did that's a little bit different than the Glow device was that we reduced the number of channels from 4 to 2, just because it's easier to feed cells with that. But we created our mold, and when we cast both the SEBS and the PDMS worked really well, that device was fine. But what we noticed when we tried to cast the SEBS was that because the mold walls were so thick, the SEBS would dry very unevenly, and the toluene, when it would evaporate also unevenly. So we were left with really thin devices that were not smooth, they were very uneven, and basically what we realized was that this method of trying to create a mold with sebs was not working. So we looked more into the literature, and what we found was that a lot of people used injection molding with sebs to create their devices, so then we decided that maybe we should try injection molding. But while we were doing that, we saw that sebs, in order to injection mold sebs, we would have to buy like a new form of sebs that could be melted into the mold. So we thought maybe we could use polystyrene, which is also, it does not absorb hydrophobic molecules, and it's also non-PDMS, since the main purpose of our device was to use a non-PDMS material. So we decided to maybe try using polystyrene and injection mold it to create a device. And since we were already creating an injection mold, we already had to create like a new CAD for the injection mold, so we also decided to modify the device a little bit based off of what we saw with our PDMS device. So our final device design looks like this. So we have like our glass slide on the bottom, and on top of that we have a sheet adhesive which is cut into shapes with the vinyl cutter. On top of that we have our bottom channel, which we cut using polycarbonate, which is clear. And we had, so initially this was supposed to be another sheet of adhesive, but we found that the adhesive didn't really stick to our polystyrene mold on top, so what we used was a spray-on layer of adhesive. We had a membrane, and then this was like our, instead of sebs, what we used was our injection molded polystyrene device up there. And as you can see, Margaret will show like better images later, but instead of having 2 channels, we reduced the number of channels to 1, and now we have large media reservoirs in addition.
So to go more into depth about our final fabrication process, this is kind of like our workflow. So initially we use a 3D printer using standard resin to print our top channel molds and our bottom channel mold, but that had some difficulties, which I'll talk about later. And then following that, we use injection molding to essentially produce this polystyrene top channel cast. And then from there, we use the vinyl cutter to ultimately use material polycarbonate to produce our bottom channel and also produce our adhesive layers. And then from there, we're able to use a spray adhesive to ultimately assemble all of the essential elements of our device, including the membrane, and the top channel, bottom channel, and we're able to produce our final device shown there. So to go into more depth about the dimensions of our top channel, this is what our final CAD was. And specifically, you can see where we hope to put our CAD2 cells and where we hope to essentially see through this top channel and have our cells laying here. And these are like the more specific dimensions. And then, so these were our original molds that we created using the standard resin that we 3D printed. So here we have our top channel mold, and then we have our bottom channel mold. And so the next step we wanted to do was essentially use injection molding for both top and bottom channels. However, we had a lot of difficulties. Injection molding is not as easy as it sounds, and since we were using a different dimension device than the original thing in injection molding, it was hard to kind of finagle our way into putting our device to actually like perfectly align where we wanted to insert the polystyrene. So these are some of our failed attempts. But eventually, after a lot of unsuccessful attempts, we were able to produce some pretty solid polystyrene top channel cast. However, the depth of the dimension of the bottom layer from before was actually too thin to actually use injection molding. So we decided to look towards a different alternative, and that's where we decided to use the vinyl cutter to essentially polycarbonate a different material that is still durable and easy to work with to produce this thinner layer here. And these are the specific dimensions that we used from our CAD. Essentially, we used the program Simulate to put in our CAD dimensions and then easily print it out, and that took like 10 minutes. So after using our spray adhesive and also a cutout adhesive sheet, we were able to assemble our final device here, and we were able to essentially achieve the novelty that we were hoping for by using a non-hydrophobic material, polystyrene. And in the future, we wanted to conduct absorption assays to officially confirm using quantitative analysis to see that there was no absorption occurring. So before we could eventually seed our cells, we wanted to prove that our device was leak-proof. And so we conducted a food dye test, and this was the results, proving that it would be fine for cell seeding. And Francisco will talk more about that.
Yeah, so we used a pretty standard protocol to seed our CACO2 cells. So CACO2 cells are from a 72-year-old Caucasian male with adenocarcinoma. And so we just used DMEM media, which was served as our defined media, and then fetal bovine serum as our undefined media, which kind of contains like a lot of serum proteins and goodies that cells need to survive. And then we included the 1% penicillin and streptomycin, which are just two antibiotics to prevent bacterial contamination of our cells. So we incubated these at standard, in a standard cell incubator at 37 degrees Celsius with 5% CO2 and 95% relative humidity. And we used just a standard passaging protocol to maintain these cells for the weeks that we do need them. And so just washing with PBS and then a 5-minute trypsin incubation to unadhere the cells from the flasks themselves. And so just kind of going back to our polystyrene material, so the flasks used in tissue culture are also made from polystyrene. And so we expected to not really have like that many issues with biocompatibility since we were basically using the same material. And so we confirmed this with the cell viability test. So we stained our cells with trypan blue. And so what trypan blue does is it's only able to stain cells that are dead because it isn't able to enter cells with intact cell membranes. And so we had really good viability results and really high concentrations of cells, so no real problems there. But then, so once we actually started seeding into our device, we loaded our cells into the top channel, and we have the top channel, where our cells go, and then we have a media reservoir underneath that they use. And so even though we had initial good cell viability when we seeded our cells, what our theory is that since none of us have ever really worked with such like small volumes of media before, it's likely that overnight, just in the incubator, due to the higher temperature, that a lot of the media evaporated. And so we think our cells just basically shriveled up and lysed after that. And so you can see here we used a DAPI stain to stain for the nucleus in our cells, and we can see that there are definitely cells in our device at one point. But we also tried to stain with actin green, and actin is an important part of the cytoskeleton, so it kind of like maps the outline of individual cells, and you can't really see like any defined cell cytoskeletons around our cells. So yeah, we think the cells just shriveled up because of that media evaporation. And so a potential solution to this would just be increasing the humidity in our, since these devices were incubated in the incubator inside of petri dishes, just increasing the humidity inside that petri dish with a little reservoir of water. And so just some future plans for our device. After finalizing our fabrication process, it's pretty efficient, and so it's really easy to mass produce our devices. Once we have like a better CAD with a mold that more perfectly fit the dimensions that are supposed to be used in the injection mold, it's really easy to mass produce these devices, and injection molding is a very high throughput fabrication method. And we still want to investigate other device materials and fabrication methods. So like Anupama mentioned, we weren't able to use the SEBs because we didn't have a formulation of the polymer that could be used in injection molding. And if we had more time, it's definitely an avenue we'd still like to explore. There's one polymer that we identified named Pratt & G, and it's patented by a company, so we would have to have some communication there and see if we could purchase some from them. But we think it would be really useful in the microfluidic device field. We'd also like to co-culture our CACO2 cells with bacterial strains, and so that would involve removing the antibiotics from our media and just keeping really good sterile technique while we culture our cells. And then we'd also like to conduct drug metabolism assays. We saw some really interesting ones that have been done in literature before, like testing drugs like acetaminophen or ibuprofen, which are like pain relieving drugs, to see how they affect toxicity of cells in the intestine, of intestinal cells. So yeah, I guess just like some reflections. So it's really important. One thing we noticed was that it's really important, like from the get-go, when you're working in an interdisciplinary team, just kind of share the knowledge that you have between each other, and very like integral skill sets that are needed for a fabrication process need to be taught to other teammates from the get-go, because otherwise you can easily get held up with maybe only one person knows how to do this process and they have like a really busy week, so then you're just kind of waiting on them because no one else knows how to do it. So if you get that out of the way earlier and multiple members of your team are trained, then you don't run into that issue. Planning and preparing for failed outcomes is also really important. I think it's like really easy to lose perspective when you're reading like a lot of these papers. You know, a lot of times it's PhD students or postdocs who have like more experience than you do in the field, and they're generally like they work over much like longer time frames, and so since scientific papers are so condensed, it's really easy to just kind of read through something. You're like, oh yeah, I can do this really quick, and it's like, well, maybe it took a PhD student like 3 months to like really finagle and like work out all the kinks in the process. Setting realistic deadlines for yourself is kind of ties into that too. Just like, since you're going to probably take longer than someone with more experience, you need to budget for that time. And then mimicking and I get transitioning to more just like things we learned about the organ on a chip field in general. We learned a lot about how mimicking an in vivo system just has so many layers of complexity to it. There's like just billions of years of like evolution that have led to humans today, and so recreating that in a microfluidic device isn't going to happen overnight, and it's important to keep that in mind. And we also learned that there are many different design paths to producing a successful device, which is kind of something unique to this class. Many times you have like a defined set of steps that you have to follow through, but with this class you kind of have the flexibility to, since it's just research, you can take any path to, as long as you achieve your final goal, which here was creating our successful non-PDMS based device. And then we also learned just how much the field of microfluidics is like still in its infancy, and how much more like there's endless opportunity to improve upon current models.